So essentially, I want to take us back to where life initially started. So we know that there was a prototypical cell that was able to grow and divide, essentially, and kind of like colonize certain areas. Now, this cell obviously then led to an entire population of cells. And in this population of cells, they didn't just really grow in a vacuum with everything that they needed to grow. They kind of like had these, what we call stressors, that existed in the physical environment. And these stressors, they had to kind of like overcome and adapt. And for them to adapt efficiently to it, they also had to sort of change their genomic code. And we know this process to be evolution, and we also understand these stressors to be known as selection pressures. Now, selection pressures then gave rise to a whole diversity of organisms in the environment, and when there's a diversity, there's also competition. And in this competition, microbes kind of found these really cool ways to overcome, or I guess like kill each other. And one of the things that they used to do is actually secrete lethal compounds that would kind of like kill off some of the others that survived, some of the others in the environment. And those that survived, we knew that they had some kind of genetic predisposition to do so, and they were able to tolerate these drugs. Now, over time, as they continued on to tolerate these compounds, they kind of evolved into a whole host and multitude of microbial organisms. Now, this process took millions of years. Like, I'm kind of like going through like a very like brief history of it, but we currently understand the kingdom of life to kind of like fall into these broad categories where we have usually like cellular organisms and then complex ones. We are lucky we fall into the complex ones, but unfortunately most people tend to think of us like this. We only see ourselves as an organism to an organism, so we see a dog, a giraffe, and like, yeah, those are living things. But most of life is actually comprised of cells. And you can see that any system, including us, broken down to its basics, actually involves cells as well. And this is what microbiologists try to highlight and push to people. Now, not only do we have this now like cellular environment happening, we also have kind of more competition. We are complex. These microbes are what we would describe as simple, but they're still interacting with us. They're still finding ways to either kill or harm us, or in some even cooperate with us, that we're starting to learn with like the gut bacteria in us. But throughout most of human history, we've had huge epidemics, such as like the Black Plague, Great Pestilence, and cholera outbreaks. Now, these are huge moments in human history, and they've killed over hundreds of millions of people. And only with like recent, well, I guess hindsight and recent understanding, we've come to realize that these diseases were also caused by microbes. We have either cases of Yersinia, Salmonella, or cholera itself causing these problems. Now, I want you to appreciate that this is how we used to die. We used to die like through these organisms killing us, and now we kind of don't. But before I get on to why we don't anymore, I want you to really take in these 2 points that I'll be carrying forward. And one is that they're interacting with us at a cellular level. You don't think of bacteria or fungi in general because they're not big, they're not punching you, they're like almost invisible. But when they attack our cells, they are attacking us. And aside from that, I really want you to know that we've kind of forgotten that it's so important. These problems that we're facing, we don't really face them anymore, and so we're like, oh, like humanity's fine, like I can afford to get a cut, I can get into a car crash, like, and, you know, light injuries aren't a worry. Now, this is because of the discovery of antibiotics, and I'm sure at this point we all know what antibiotics are. They're like these like life-saving molecules that do something, you know? Our doctor says we're sick, we just take them. That's not really the case, and I want to show you why. So let's take an example of a cell right here. I'll show you like how antibiotics tend to work. So we've either discovered or invented compounds that can affect the cell wall and cell membrane, stop the actual bacteria or fungi from replicating, interact directly with components in the cell, such as like proteins or lipids, to prevent future functions, or even attack the ribosomes, which kind of like synthesize everything the cell needs to survive. Now, this is really great and really amazing, and by affecting the metabolism of these cells, we tend to then cause cell death. Now, as I also described earlier, cell death is a process known as natural selection. And through natural selection, these organisms actually find ways to overcome these pressures and actually adapt. Now, on the other hand, whilst we have these molecules that have given us all of these advancements, these microbes have also found ways to actually resist them. It usually starts with a change in their genome that is accompanied by functions such as making the cell wall impermeable so that they can't enter the cell, or even just changing the target that the drug is supposed to bind to so that it doesn't bind to it anymore, or even just kind of like seeing and sensing that there's a compound in the cell, and it's like, oh, this is doing damage, let's just take it out. And the thing is, these bugs have found numerous ways to combat most of the compounds that we throw at them just because of the fact that it's selecting for only those that can survive. And this ends up being a huge problem because microbes can propagate very quickly. They spread like nothing. The generation time is of 18 days, whereas like the human generation time could be like 18 years, right? And so what I want you to also realize is that we have a huge problem emerging with this. And the WHO actually predicts that with the spread of bugs that are resistant to drugs, we would have 10 million deaths by the year 2050. And yes, well, you can see that this is localized very heavily towards developing nations. We have issues where even here on our developed world, this would kind of come in and also spread. You can imagine that a bug that is found in India can easily come to the States or easily go to Australia. Like, we know how globalized society is. Now, I just kind of spoke about bugs and how resistant they are, and you're probably wondering, well, why are we fine? And for the most part, we're fine because of the fact that we also have a secondary protection system known as our immune system. And they kind of like our immune cells do a good job of keeping us alive for the most part. But these important pathogens also have ways to circumvent the traditional roles of our immune system. And I'm not going to take you through everything that does everything, because that's literally a field. But I'll tell you about the pathogen that I work with. It's Candida albicans, and it's a fungal pathogen. Now, I'm about to show you a small video. And Candida albicans are the cells highlighted in green, whilst our immune cells are highlighted in yellow. And I want to show you how they, once they become drug resistant, also how they interact with our immune system. Now, before I also begin, Candida albicans has this capacity to what we call filament. It forms like these long, like protruding versions of itself to burrow into our tissues and actually kill more cells that way. Now, what we can actually see here, they start to filament, and besides just regularly filamenting, the yeast cell that's inside of the immune cell actually filaments outside of it. It breaks the cell open. And this is insane. Like, this is how our body is supposed to naturally defend itself, and this organism is showing us that it doesn't care about that. And there's so many microbes that essentially function in the exact same way. So at this point, again, I realized I just said, oh, like they're resistant to all the compounds that we make. We have an immune system. A lot of them can like circumvent how the immune system works. So what's the problem? Now we can appreciate why the WHO says by 2050, 10 million of us will be dead annually. And it's quite simple. What we're going to do is just kind of take away what actually makes them a pathogen. The kingdom of life, they're mostly cells. Some of the cells have adapted to killing or harming other cells. And if we can stop that process, then they just continue living. And you might think, okay, if we stop the pathogen from being a pathogen, that's like a pretty smart idea. Why aren't we doing it? And the question is, we never really had to. I told you before in human history, they caused millions of deaths. We just knew we had a problem. We can kill them this way. Let's kill them. And nature's found this way to overcome this through evolution. And now we're kind of like left with the original problem. And humans have kind of coming up with these cool, inventive solutions. And I want to talk about that just for a little bit. So with the pathogen I'm working with, albicans, we can see it like under regular conditions filamenting here. But there's a compound, for simplicity's sake, we'll call 902. With the induction of 902, we can see a complete abolishment of albicans and its capacity to filament. That's huge because of the fact that it's its main virulence factor, the fact that it can burrow into our cells to cause even more damage. And we also see that when we try to mimic the human body, yes, they continue to filament, but when we put them in these exact same conditions, and we also add the compound 902, even when it's trying to mimic being in the human body and filamenting, it can't. And that is something that's super crazy and super new for us because of the fact that we are really now looking at specific or precision therapeutics. We're looking at targeting specifically the pathogens that exist within us. And I know I'm saying pathogens and I'm only talking about one, but I want to show you guys that we're also working on Clostridium infections. We're looking on anthrax. We're even looking, sorry. So this is another part that I just kind of wanted to just briefly go over. And it's that this compound, 902, kind of like completely inhibits the cell from forming these like really dense networks. And besides from just forming these dense networks, it can actually increase the survival rate. And that's something that I kind of want to go on to when I'm telling you that we're solving for all these big infections, even drug-resistant ones, such as tuberculosis and staph infections. And what we can kind of see by all this research that's being poured in is that there's a consistent survival rate with all these organisms when we stop these bugs from becoming virulent. Now, that's important because before, again, our prior understanding has been that we've had to kill them. And we've had to kill them just because they're killing us. But now we're seeing that we can treat them, let them be cells, and we still survive. And this, of course, are results that we're extrapolating because of the fact that we usually use like mouse models or worms, and we're really moving into an era where we can sort of treat humans without killing these cells. Now, the really cool aspect of this, I'm talking about like a lot of cool aspects, and I guess it's because I'm also showing like research that's only been done in the last decade, the last bit of the decade. Most people aren't aware of the fact that we're trying to basically disarm pathogens from being pathogens, right? You're still thinking, oh, tuberculosis is bad, we should kill it. Now, why aren't we just spending time inventing more drugs? Well, we're not doing that because, as I said earlier, when we invent different drugs, they find new ways to just overcome it. It's kind of like a tireless race that we're realizing we'll quickly lose if we keep it up. So we are creating these compounds that can stop them. Now, whilst we're stopping them, you might be asking, okay, I just spoke about them evolving resistance. Can't they evolve resistance to these compounds? So that's actually what a pretty similar study kind of like looked at. This is not 902, but a compound very similar to it. We see that it's stopping filamentation with just micromolars of the solution. Then what they did, looking at a classical strain of these guys, of this pathogen, is they gave it just a little bit of the compound. And they grew them over a couple days in the presence of the compound, seeing if it still had the capacity to filament. And then after 40 days, they just gave them a huge amount, just to see like if any that are resistant will still survive, or if we see any drops in the level of the amount of cells present. We don't see that. We don't see any evolution of resistance. This compound that can initially inhibit hyphal formation, even when we try to get these microbes to form hyphae, they're still not forming it. And this is because of the fact that they're not dying, they're just stopping from having an action. And that's a really good point, and that's a really good thing. They can't really be any natural selection taking place. There's no selection pressure, nobody's dying, right? They're just living, and they're not avirulent. And what's even cooler is that this is a strain that is known to be hypervirulent, very drug resistant, and it kills mice faster than you can actually imagine. And even with that, we see again, absolutely no resistance. And this is huge. This is like the biggest thing that's ever really happened to us in the longest while. We see this war, and we have this war that we've been losing, and suddenly we have this kind of like precision therapeutic that we can use to stop not only microbial infections that regularly exist, but microbial infections that are drug resistant. We have people that are still dying from all of these things. And yes, I can acknowledge that in the data that I showed you, they weren't always 100% success rates. Now, this is where the funny thing comes into play, because this was probably one of the first antibiotic talks given that doesn't say, stop using antibiotics. What I'm actually trying to say with you guys is that we can use these compounds to treat infections, but antibiotics also still have a place. There are certain infections with certain pathogens that are far too invasive or too deep-seated that when doctors actually get to them, like these compounds that's supposed to stop the virulence factors don't. And that's when we're supposed to use antibiotics. Antibiotics were always supposed to be the last resort. And honestly, not to be preachy, but one of the really fun and cool things happening here is that there's so many groups happening, like studying this all across the world, and they're seeing the same results. We can successfully save people, and then we can also save the ones that can't be treated with these infectives, these anti-infectives we call them, with antimicrobial drugs. And they have huge efficacy, and they work, and we are now starting to live again. Thank you.